So, today we are going to talk about T cell development in the thymus. I'm going to make some a quick run through of some points from B cell development because we're going to do a little compare and contrasting. So that's why I have some B cell development stuff up on the board. I have two general pieces of information. Got it? Two general things to say about the information that you're going to hear today. Just because this is one class period where some of the material evokes strong reactions. So number one, please remember that peripheral tolerance exists. Number two, please remember that the immune system originated through the process of evolution and only had the things that evolution had out there as tools in order to make the immune system. So there are parts that you're going to hear today and you're going to be like, no. That's a bad way to make it work. And that was what we had options. Those were the options we had. So when you get to that slide, or when we get to that part, I promise that's what evolution had to do. So we talked about, we've been talking about the T cell receptor. We talked about kind of the general ideas of T cell development and VDJ in T cells, and the fact that it looked pretty similar to what we saw in B cells in terms of that VDJ recombination. But that all of the parts of T cell development happen in the thymus, as we talked about last time. And just as a brief overview, here is B cell development. So you can remember we started out with the pro-B cell. It did D of the heavy chain to J of the heavy chain rearrangement, then V to DJ. Then it had a heavy chain. And it put the heavy chain on the surface along with a surrogate light chain and called it the preBCR. And once it had the preBCR on its surface, we called it a pre-B cell. That pre-B cell did some signaling events through the preBCR, allelic exclusion, survival. It turned off the surrogate light chain, it proliferated, and it then did light chain rearrangement so that it could make a light chain. Then once it had a light chain, it put that on the cell surface, and we had an immature B cell, which I forgot to write. And we did our testing for self-reactivity in those immature B cells, so that central tolerance mechanism. And if you recall, in our central tolerance mechanisms, there was a problem with B cells and central tolerance. There was a place where we were like, oh, that's a loophole. There's definitely some autoreactive B cells that make it through this process because of this loophole. So what was the loophole? What was the issue that we saw with B cell development? Yep.
All of the cells that the B cell reacted for were found in the antibody.
So it's not so much the cells, it's the antigens. So every antigen, every potential antigen in your body is not found in the bone marrow. And so we don't have the ability to test against all antigens in the bone marrow. And as I told you, happily, we have peripheral tolerance mechanisms to help deal with that. So today we're going to be kind of going through the parallel versions of a lot of this for T cells. T cells start out their development from that same common lymphoid progenitor that B cells do. It's just that those progenitors go to the thymus to undergo their developmental processes. That common lymphoid progenitor came from the hematopoietic stem cell. Apparently the hematopoietic stem cell talk has been rescheduled for October 21st. So if you wanted to go, that's when it is. And we can see kind of some slightly similar stuff. And so if we went at a super big picture level, this is not entirely how we usually talk about it, but if we went at a super big picture level, I'm going to show you here how we might think about T cell development. So what you will notice is that basically what I did here is that I took all the places where I had BCR and I erased the B and I wrote a T for TCR. And again, if we were at a very big picture level, this is what we would see for T cell development. Unfortunately, T cells have a couple other things they have to achieve during their development, so it's not quite this straightforward. And also, unfortunately, the most unfortunate part, immunologists are obnoxious, and we rarely use the terms pro-T cell, pre-T cell, and immature T cell. We instead have different names. But it's the same set of stuff happening, it's just we put different names on them. But it, in fact, really does look like this. Every so often, you will hear people talking about pro-T cells and pre-T cells, although we end up using some slightly different names for them most of the time. Yeah? Yeah, V to J. I was writing quickly. There. Okay. So we tend to talk about T cells that are undergoing development based on their expression of certain proteins on their surface and where they end up falling on two different types of flow cytometry plots. So the most common plot that people think of when they think of the thymus, like when I think thymus, this picture goes straight into my head, is this picture of cells of the thymus being run with CD4 and CD8. And we have 4 different populations of cells when we look at this plot. We have one population of cells that are called CD4 minus, CD8 minus. And when we are talking about the thymus and a CD4, CD8 plot, we often refer to those as double negatives, or DNs. We have cells that are CD8 positive and CD4 minus. We usually refer to them when we're talking about T cells and T cell development as the CD8 single positives. We have CD4 single positives. They're CD4 positive, CD8 negative. And we have cells that are CD4 positive, CD8 positive, or double positives, or DPs. And so we tend to use names like that instead of that pro-T cell, pre-T cell thing. And in fact, the cells sort of progress as are indicated here by the arrows. They start as double negatives, and then they become double positives, and then they become single positives, as the arrows say. That's something that we will get to as we're talking through this process. And so we have to figure out their sort of choice of CD4 versus CD8 in our overall scheme. It has also been realized that the double negative cells can be further subdivided. So there are multiple different types of double negative cells. So you could take those double negative cells and you could look at their expression of 2 other proteins. One is called CD25, the other is called CD44. It turns out that at the beginning, the cells are CD44 plus, CD25 minus. They then become CD44 plus, CD25 plus, then CD25 plus, CD44 minus, then CD44 minus, CD25 minus. And so we call those populations DN1, DN2, DN3, and DN4. And so this cell actually progresses through these stages. So it's a DN1, then it's a DN2, then it's a DN3, then it's a DN4. Then actually after it's done being a DN4, it's a double positive. So all of these are kind of a subset of what's happening down here. You can see the thymus plot and sort of what it generally looks like on the left. For a variety of reasons that sort of involve an inside joke, my graduate class decided that the thymus bird was our mascot. And we used to put the thymus bird on everything. So that's the thymus bird. So we use these names, DN1, DN2, DN3, DN4, DP, SP, or double positive, single positive, instead of pro T cell and pre T cell, but we're going to see the exact same events happening that we've seen before happening. So our first couple of steps that are going to happen are going to be in the cells that are either DN1s or DN2s. And you can see your textbook lists, shows this nice little picture of the DN1 and the DN2, as well as having a table that describes exactly what's going on here. And so the DN1s are the cells that are coming in to the thymus and sort of starting this process. They will start rearrangement, and DN2 will do the rest of the rearrangement. And so you can see we kind of had two steps in pro B cells. Here we actually separate them out in T cells as DN1 and DN2. And so our double negative 1s and double negative 2s are basically doing what we thought of last time as pro T cells. As a result, you can realize that while they do have CD44 on their surface, they may or may not have CD25, depending on if they're a DN1 or a DN2. They don't have a T cell receptor because they don't make it yet. And so they won't have it on their surface. There's one other thing I want you to notice from this table about what these cells are doing with their VDJ recombination, which is that if you look at the DN2, DN2 is actually doing 3 different rearrangements at the same time. DN2 is making some gammas, is rearranging gamma, rearranging delta, and rearranging beta. So it's basically everything except alpha at this point. In fact, this cell can still decide to be a gamma delta or an alpha beta. The cell has not yet decided if it's going to be alpha beta or gamma delta, and it is doing rearrangements of chains to allow it to go down both routes. It has not made the choice yet of which route it is going to do. We are currently doing all of this in the thymus cortex, which was the region that has a lot of cells. So the cells sort of come in right at the border between the two areas, and they start doing these steps in the cortex. So we're in the thymus cortex doing these steps of rearrangement. So you can see our DN2. It's been doing these heavy chain rearrangements. So what do you think is the first thing that makes a cell a DN3? What is the first big unique factor that makes you a DN3? Yeah. The presence of a heavy chain, which you could say put on the surface of the cell as, I don't know, a pre-TCR instead of a pre-BCR. So our DN3s become sort of the first cells of that are expressing a pre-T cell receptor and are able to start doing some of these processes. Yep.
So if DN1s and DN2s, you combine gamma delta and beta,
Uh-huh.
why is it called the existence of a heavy chain? Couldn't it just be an existence of any one of those chains?
Yeah. Well, we're going to get into lineage commitment, and so you can kind of start to see it there. Okay? So our DN3 is now unique because it has that pre-T cell receptor on its surface, where it has that potential beta heavy chain. As I said, we're going to get into the gamma delta part in a bit. But right now we've got perhaps that beta heavy chain that can signal. Now when we had our receptor in B cells, when we had our heavy chain on the surface, and we were looking at signaling through that heavy chain, was our heavy chain signaling by itself in B cells? No. What was it doing? What happened? Yeah, Nick? we had a surrogate light chain. So we had sort of a invariant chain that bound to the heavy chain and kind of allowed it to be on the surface as part of the pre-BCR. We have a similar surrogate light chain or a similar type of protein in T cells that is part of the pre-T cell receptor. It's only one protein, and it's known as PT alpha. Some people will refer to it as pre-T alpha. So I've actually seen it done as either pre-T alpha or PT alpha. And so we have this surrogate light chain-like protein called PT alpha, pre-T alpha, that will bind with the heavy chain. And in fact, CD3 will be expressed at this point, so we can test out that heavy chain and make sure it is a functional cell. And then we are going to have a number of different signaling events happen. One thing I will also mention is we asked the question when we talked about B cells about how we got signaling through the pre-B cell receptor. Because if you remember, it takes a heavy chain and a light chain together to bind antigen. And we only have a heavy chain now. We don't have a light chain. So how the heck can we bind antigen? And we talked about how maybe like surrogate light chains bound to each other, and that led to some signaling. We actually are relatively sure that pre-T alphas can make nice little dimers like this, and again, lead to signaling. So we don't have to have antigen as a part of this. It's just, can we get a functional heavy chain protein on the surface? So we can have a number of different signaling events that are now going to happen because of this pre-T cell receptor on the surface of our cells. Yep, Mark. It just knows that it's a protein that didn't have like a stop codon, or that it's a full protein. So again, we are not yet thinking about antigen binding. That will be a step that we deal with later. Okay. So we can see a number of different steps that will start to happen as a result of signaling from that pre-T cell receptor. And those include things like survival. Those include further allelic exclusion. That includes turning off transcription of the surrogate light chain, pre-T alpha. That includes proliferation, and that includes making the, starting the light chain or alpha chain rearrangement. You can also see kind of this same information shown here. What you will notice is where we draw that line in terms of which of these things are in a DN3's job and which of them are a DN4's job actually disagrees in different parts of the book even. It's like really a gray line. So here, like these events are DN4 and not so much DN3. But here they're DN3 and not DN4. And so exactly where we're going to draw the line between DN3 and DN4, don't get super hung up about. I actually tried to like make sure I was correct and read your textbook and make sure I knew before I came in here. And in fact, signaling at the DN3 stage leads to, and there's a sequence of 6 things. One of them is becoming a DN4 and the other one is becoming a DP. So it's not my fault that it's confusing. And so all of these steps are really happening in DN3, DN4, and you will often see heavy chain or light chain rearrangement as being listed as also part of double positive. And so the lines here are a little bit tricky. You can imagine it would be really hard to go into a thymus, look at these cells throughout their process and figure out exactly what they're doing where, sort of in vivo. And so you can see why this distinction might be a little bit tricky. And so you can also see that this figure says we're turning on the expression of CD8, which is actually what makes you a double positive. So it kind of just like skips double negative in general, DN4 here in general. So we go through all of these events in kind of DN3 and DN4 and eventually become a double positive. And so you can see this is all very much happening in the cortex. And so all of these sort of big time VDJ steps are happening largely in the cortex. And when we're going to get to the central tolerance things, the central tolerance steps are going to be really the medulla steps for T cell development. Of course, you can see here that we still have this issue about the gamma delta T cell and how we can become a gamma delta T cell as opposed to an alpha beta T cell. In those early double negative cells, we do tend to see rearrangement of beta, gamma, and delta. If the cell finishes beta first, beta ends up on the cell surface, and signaling through it will start to allelically exclude further rearrangements and will stop rearrangements of gamma and delta. If the cell finishes gamma and delta first, then that leads to allelic exclusion and we don't rearrange beta. And so some of this is based on timing in terms of which chains are finished first and which ones make a functional signal. So you could imagine that if we rearrange beta, but we don't get a full protein, there was a stop codon, it can't signal to stop. And so maybe we keep going with some of the others. Yeah?
So why is it that both gamma and delta have to be made in order to do that alpha and beta
Let me go through a couple more slides, and then I'll come back to that, okay? And so you can see this process sort of happening here, where our cells are kind of, as DN3s, either getting a signal through their beta chain or through gamma and delta to either then further their development into gamma delta cells or to further their development into double positives and then eventually single positives. This is all based on the strength of signal that they get through those receptors. So it turns out that the signal that comes through beta and just beta with the preTCR is kind of weak. See, weak. Whereas the signal that you get from having both gamma and delta is pretty strong. And that signal strength tells the cell to do a different program. So if the cell gets a strong signal, it says, aha, I'm going to do the gamma delta program. And if it gets a weak signal, it's going to say, aha, I'm going to do the alpha beta program. And so part of the reason that you need both of them is you need to be able to get that strong signal to differentiate to the gamma delta program. Okay? Does that make sense? All right. The cell can, what you will notice here, is that the cell still once that cell starts to go down the choice of being an alpha beta and makes a beta, it still actually can switch to being gamma delta up until the point where it makes its alpha chain. Once it makes its alpha chain, it's a point of no return. If the cell gets a strong signal, if it starts the gamma delta program, it's on the gamma delta program. If it's on the alpha beta track, it could still change its major until it makes alpha. Once it makes alpha, we're done. We don't get to choose, no more choices. And the reason here is because alpha rearrangement deletes delta. And so now when we do an alpha rearrangement, we have actually gotten rid of the genetic information to make delta. And so there is no more choosing of the gamma delta path anymore. And so it's really all about the strength of signal that the cell is getting through this, this process. And so we're largely going to follow the alpha beta T cell through the rest of this process, where it becomes a DN4 and then starts to turn on CD4 and CD8. To become a double positive cell. And so the other signal that we get here is a signal that says, okay, transcribe and translate CD4 and CD8 so that you can become a double positive cell. So with our double positive cells, we now have to do some testing. We have to test whether or not we have a functional receptor, and we have to test whether or not we're self-reactive. There is one other test that our T cell needs to deal with. So we've got, do you have an actual protein as your receptor? Are you self-reactive? But we also have to make sure that this cell is responding to self-MHC. And so this is where the T cell gets MHC restriction as well. And so if a T cell develops and it does not bind self-MHC, then it is basically going to be a useless cell, and we don't want that either. And so we've got a lot of different processes of testing that our double positive cell has to go through. I want to go back for a second and point one thing out. This one. If you look at this flow cytometry plot of the thymus, notice that the vast majority of cells in the thymus are double positive cells. Cells spend a while in the double positive stage, and we have a lot of them. And we have relatively few CD4s and CD8s. The vast majority of double positive cells are going to get killed. In fact, I believe the number is 95% of double positives die. And so all of this testing that we're going to do is going to involve a lot of cells failing. And so making sure we have a functional protein, making sure we have the ability to recognize self-MHC, and making sure we're not self-reactive ends up being a pretty big test, which is one of the reasons why we have to make so many double positives. So you could imagine we're going to proliferate a whole lot more here. This is also why we see big differences in the cortex and the medulla in terms of how many cells are there. Because in the medulla, we're doing the testing. We have fewer cells because we've gotten rid of a lot of them. In the cortex, we're basically making all the cells and having them divide before we do these strict types of tests. And so now we need to test those double positive cells. We have, and so these types of testing are going to be a big part of the central tolerance process, where we get rid of the self-reactive cells during apoptosis. But with T cells, we often think about two different processes that are happening to them here. One of them is called positive selection, and one of them is called negative selection. And so our T cell has to deal with two different types of selection here. So let's imagine that you were thinking about being a T cell. Would you like it, do you think, if you were negatively selected? Would you like to be negatively selected? You're shaking your head no. Why not? That sounds like a bad thing to have happen. You don't want to be negatively selected. You don't want someone to point you out and be like, you're bad. And that's kind of what negative selection is. And so if we have a cell that is negatively selected, that's bad. And in fact, the cell is going to undergo apoptosis. Okay, now let's imagine that you're a T cell. And so if you're not negatively selected, then you're sort of in the middle, right? So let's imagine you're a T cell, and I tell you that you could be positively selected. Would you like to be positively selected? Yes, that sounds good. That sounds like a nice thing. In fact, positive selection is basically like that pat on the head. Remember how I told you that B cells were needy? T cells are needy too, and positive selection is, you did a good job. And so we can have cells that are positively selected. They grow up to be T cells. We can also have T cells that do not get positively selected. Sometimes we refer to this as failing positive selection. Would you like to fail positive selection? No. What do you think happens to T cells that fail positive selection? They die. And so these cells also undergo apoptosis. And I even know how to spell them. And so our T cells undergo both processes of positive and negative selection. And we're going to go through them more on the next few slides. But this is generally referred to as the Goldilocks model of T cell development. You don't want to have too much, you don't want to have too little, you want to have just right.
Hello. The rest of the T cell development lecture is basically solid static. So I'm re-recording the second half of it after the fact so that you can clearly hear what I'm saying. I apologize if this doesn't perfectly match with your notes from the lecture, and I apologize for you not getting to hear student questions, but hopefully you'll actually hear the content unlike the way that you would have on the previous recording. In the part that you just heard, I had just talked a little bit about negative and positive selection and introduced the Goldilocks model of thymic selection. We then move on to this slide where I'm talking a little bit more about the Goldilocks model of thymic selection. If we have a cell that has very low or possibly even no affinity for anything it sees in the thymus, then that cell is going to get no signal. That cell is going to fail to be positively selected. And we can assume that that cell doesn't bind to MHC. Because that cell gets no signal telling it to live, it's going to die. We usually refer to this process as death by neglect. Or sometimes you will hear about somebody talking about ignoring the useless. If the cell has very high affinity, that cell is going to be actively killed because that is likely a self-reactive cell that is going to be very dangerous in the periphery. So those cells are also going to die by apoptosis, although it's an active deletion, and that process is going to be called negatively selected. You can imagine that you would not like to be negatively selected. That sounds like a bad thing. If we have a cell that has kind of medium affinity in the thymus, then that cell is going to get a signal in order to keep living. It's going to keep developing, and it's eventually going to become a T cell that we see in the periphery. That sort of pat on the head that the T cell is getting that gives it a signal to survive is known as positive selection. So a cell does not want to be negatively selected, but a cell doesn't want to be positively selected. The cells that I told you about before that die by neglect are often referred to as having failed positive selection. Sometimes people will talk here about killing the harmful, which is negative selection, keeping the useful, which is positive selection, and ignoring the harmless. You can also notice that there is the vast majority of the cells in the thymus die. This is one reason why we need to have a lot of proliferation during some of our T cell development steps, because we are eventually going to have so many cell types die. This is why we have so many double positives, but so few single positives will end up coming out of the thymus. And this is also why the part of the thymus where these processes are happening ends up having fewer cells in it, being sort of lighter, easier, different to see under a microscope because there are so many cells that have died, there are fewer cells. You'll also notice that there is a fourth outcome for T cells here that have high affinity. Sometimes we talk about very high or high affinity as being negative selection, low or no affinity as being neglect, and sort of low intermediate as being positive selection. There is also a high intermediate option that is shown here, and I've whited out the details of what happens in that option because that's something that we're going to talk about later on in this semester. And so here you can see sort of another example of that process. You can see that the cells will first undergo positive selection, where they, if they get no binding to and get no signal, then they're going to undergo neglect, they're going to die. And if they have moderate or strong binding, they're going to get that positive selection signal and they're going to live. However, then we're going to see additional testing for that either positive selection or a very strong signal of negative selection where the cell is going to eventually die. This process is largely happening in the medulla, and we're going to see interaction between the T cells and cells of the medulla. And so you can see that here as well. Many of the original processes of VDJ rearrangement that are happening for our cell, where the cell is going to become a double positive, will happen in the cortex. The cell is going to start undergoing its selective processes in the cortex, but the medulla is really the location where we see sort of our central tolerance induction and our T cell selection processes in our double positive T cells. And so you can see the Goldilocks model listed here as well. Unfortunately, there's a really big problem that we can see with the Goldilocks model. So there are a couple of issues, in fact. So there are a couple of issues that we have to think about when thinking about the Goldilocks model. You can tell from this and from other things that I've told you about that our T cells that are developing in the thymus must be interacting with some of the cells of the thymus in order to get some type of signal. T cells and the T cell receptor only see antigen when bound to MHC molecules. MHC class 1, of course, is expressed on all nucleated cells, and so this is easy. All cells of the thymus are nucleated cells, and so all of those stromal cells will be presenting on class 1. That will be an easy part of this. But what about class 2? How is there class 2 in the thymus for presentation of antigen? Well, if you recall, when I told you about MHC class 2 expression, I said that there are 3 types of professional antigen presenting cells: dendritic cells, macrophages, and B cells that present on MHC class 2 all the time. I also mentioned that there are a few types of non-professional antigen presenting cells that can sometimes present on MHC class 2. The only one that I really pointed out were thymic epithelial cells. So cells of the thymus are able to also express and present on MHC class 2, thus they can support the development of T cells, especially CD4 T cells. However, there's another problem that we can think about when we think about the Goldilocks model of thymus selection. If you remember back to B cells, there was this problem of, oh wait, there are only proteins that are expressed in the bone marrow available for testing for self-antigens for our B cells. And that is a little bit of a problem. And so you might ask, well, are there antigens available in the thymus? What types of antigens are available for testing in the thymus? But there's actually one other somewhat related problem in the thymus. I'm pretty sure in the actual lecture I talked about this a little earlier, but it's a key point that we have to understand when thinking about thymic selection. In the thymus, the only antigens that are available are self-antigens. So, when our T cells are trying to get a signal, they're trying to get that intermediate signal for positive selection, the only antigens on which our T cells can be selected in the thymus are self-antigens. So the idea is that if these T cells are binding weakly to self-antigen, they are probably able to bind MHC, and perhaps when there is a similar antigen, maybe with an amino acid or two difference in the periphery, these cells might be cross-reactive and able to respond. But the end result is that all of our T cells that make it out of the thymus had to get positively selected, and thus they all had to have some type of low affinity for self-antigen. As a result, every T cell that comes out of the thymus is a little bit self-reactive. Hopefully, it's not going to make a very strong response to self-antigens, but it is able to make somewhat of a weak response. It had to be able to do so in order to get positively selected and leave the thymus. And this is sort of the only way we can really have our cells be MHC restricted. If a cell is not able to bind MHC, it gets no signal at all, but we don't have sort of other types of antigens to test on in the thymus. There's still that question of what about the antigens that are in the periphery. As we said, B cells, when they are developing, don't get tested on peripheral antigens. So there are probably some B cells that make their way into the periphery that are going to be self-reactive because their antigen was not present in the bone marrow for negative selection. You could imagine that this could be a real problem with T cells, since we also have the problem that every T cell is a little bit self-reactive. Fortunately, it turns out that actually all antigens are present in the thymus. So basically, every gene in your genome is transcribed and translated in the thymus so that all self proteins are available as antigens for T cell testing in the thymus to do negative selection against all of those possible antigens. We often think about some types of proteins as being tissue-specific proteins. They might be proteins that are only transcribed and translated in the retina or in the ovaries. However, in the thymus, we also transcribe and translate all of those proteins, meaning that all of the antigens are available for negative selection in the thymus. This is done via a protein called AIR, or the autoimmune regulator, which leads to the transcription of all of those tissue-specific proteins, allowing thymic medullary cells to make all those proteins and delete these self-reactive T cells. AIR alters the transcription of many of these genes, and for a long time, people have kind of thought about AIR as a transcription factor. However, the experiments that would be needed to demonstrate that AIR is in fact a transcription factor have never actually been successfully completed. So there are lots of ways that it seems like a transcription factor, but no one can actually show that it is. Perhaps it is regulating some other type of transcription factor that we do not yet know. These data are from one of the papers that first described AIR. Here, the authors were looking at genes that were whose transcription seemed to be controlled by AIR. You can see that many of them are red, and they are genes that seem to be transcribed only in one specific tissue as well as in the thymus. This includes things like oxytocin from the brain, salivary protein 2 from salivary glands, preproinsulin, things like that. You can see that many of the others are also tissue-specific-ish genes as well. And so this was sort of this idea that showed that AIR was responsible for making sure tissue-specific genes are turned on in the thymus to ensure that those antigens are present for T cell selection. AIR was first described in a group of patients with an autoimmune disease known as APSED, which stands for autoimmune polyendocrinopathy, candidiasis, and ectodermal dystrophy. In these patients, AIR is not made, which means that many tissue-specific antigens are not made in the thymus, and so many different types of T cells are able to go into the periphery and be self-reactive that shouldn't actually be there. They escape into the periphery. As a result, these patients don't just end up with one type of autoimmune disease. They tend to have multiple autoimmune diseases affecting multiple organs because they're having such a problem broadly with central tolerance. Each APSED patient has a slightly different set of autoimmune symptoms. You can see that the majority, 85%, have hypoparathyroidism. Many have adrenal failure. Many of the women have ovarian failure. Some will have diabetes. Some of the men will have testicular atrophy, and on and on and on. It's also been noted that all of them, 100%, have candidiasis, which is a recurrent yeast infection, and many of them have issues with their fingernails, nail dystrophy, which is shown here. So each patient has a slightly different set of these types of conditions based on which T cells they were unlucky enough to have sort of make it out of the thymus. There are mice that are deficient in AIR. Those mice do not have as many autoimmune symptoms as we might expect, although they have very, very, very messed up salivary glands because they seem to be having a lot of autoimmune attack at the salivary gland. It's weird. I don't know why. So here you can see the details of the T cell development and selection locations that we've been talking about. We are still talking about double positive cells. Those double positive cells produce a T cell receptor in the cortex and start the selection there. Remember, the cortex is relatively densely packed with cells, while the medulla is much more loosely packed. The cells will be moving into the medulla to start selection, and since many of them will die and we won't see any proliferation there, we will tend to see fewer cells in the thymus. And so you can see that same process here as well, where our cells are going to sort of move their way out of the cortex into the medulla and finish up development. The final part of this process is that our cells are going to become mature T cells. Mature T cells are the single positive, CD4 positive, or CD8 positive cells that you can see here. These cells are now ready to act as T cells. They have not yet left the thymus, but they're getting ready to leave to go out into the periphery to make some responses. And so ideally now when these cells get a signal through their T cell receptor, they'll proliferate, they'll survive, and they'll start to do some sort of function. So you can see those mature T cells here where they now are able to be single positives and leave the thymus in order to do their effector functions. As a result, these cells go from being double positive CD4 positive CD8 T cells to being single positive either CD4 positive or CD8 positive T cells. And so they have to actually sort of choose which of those two proteins, CD4 or CD8, they want to keep expressing. There are a few models of co-receptor choice that have been proposed, and the data are not clear on exactly which model of co-receptor choice happens. There are some ideas that there is this instructive model where the cell starts out as a double positive. It sort of sees which one it gets the better response from and then keeps that one, either CD4 or CD8. And then goes forward. There's also a model that's known as sort of the stochastic model, where the cell randomly either upregulates CD4 or CD8 and then tests to see whether it is able to get a signal through the protein that it has made. If it does not get a signal, it undergoes apoptosis. And there are other types of more complex models that have been proposed. So somehow that cell is going to sort of choose a particular co-receptor, although the specific details of that are not completely clear. And so at this point, our cells are going to be able to leave the thymus and move to secondary lymphoid organs where they can make a response in the periphery. These are some figures of what we might see if we wanted to look at T cells in the periphery. You will notice this looks different than when we look at T cells in the thymus. We don't see that sort of characteristic little thymus bird. So if we're talking about either the lymph nodes or the spleen, those tend to be the organs I reference the most often when I'm thinking about T cells in the periphery. So if I ask you about one of those on an exam, I'm trying to get you to think about the periphery. And we can look at the CD4 and CD8 flow cytometry plots as we see here. The first thing that you should notice is that there are very few double positives. In fact, really, you should think about this as there are no double positives. You can imagine drawing no population there. You can see that there are lots of CD4s and there are lots of CD8s. So no double positives, lots of each of the types of single positives. There also can be a question of whether or not there are double negatives. In these plots, you see double negatives. In these plots, we are looking at all of the lymphocytes in the lymph node or all of the lymphocytes in the spleen. So those double negatives might be B cells or NK cells or something like that. If you were to perform gating on CD3 so that your analysis was just looking at T cells, you would only see the single positive cells. You would see no double positives because it's the periphery, but you would also see no double negatives because you've done gating. You're looking at only T cells and you're really only going to see those single positive T cells in the periphery. So those are types of things you should be thinking about when trying to represent peripheral T cells. Now that our T cells are in the periphery, they're finally able to start making responses. And we're finally going to be able to actually look at the process of some adaptive immune responses. Sometimes we look at T cells in the periphery to help us understand what we might be seeing as far as development in the thymus. So we expect in the periphery to see CD4 T cells and CD8 T cells, both single positive types, but if we perhaps see something different, that might indicate that something strange was going on during T cell development in the thymus. So one of the ways that we might look at this is we might use mice that are either missing MHC class 1 or MHC class 2. Here we're looking in the thymus, and you can see that in the thymus of control mice, there are double negative cells, there are double positive cells, there are CD4 positive cells, and there are CD8 positive cells. If you were to have mice that were deficient in MHC class 1, you can see that the double negatives would be present, the double positives would be present, and CD4 positive cells would be present. MHC class 2 would still be present. You'd be able to develop a CD4 positive cell, but CD8s would be missing. Of course, class 1 binds to CD8, and so that's the link there. And with an MHC class 2 deficient mouse, you see that the CD4 positive cells would be missing, but the double negatives, the double positives, and the CD8s would be present. And if we looked in the periphery, the class 1 deficient mouse would have CD4s but not CD8s. The class 2 deficient mouse would have CD8s but not CD4s. We can also imagine generating mice that are T cell transgenic mice. These are similar to the B cell transgenic mice that we talked about earlier when we talked about B cells. So we might take a mouse like the H2K background mouse that's shown in the upper right-hand corner, and we might find some T cell in that mouse that we really like, as is shown here. Whatever that T cell is that we picked will be either a CD4 or a CD8. It will have some kind of T cell receptor that binds to some MHC plus peptide. In this case, it is a class 1 MHC that has an influenza peptide, and it is one of the H2K MHC that is recognized. And we can isolate the alpha and the beta T cell receptor genes from that mouse. We can then insert the pre-rearranged alpha and beta genes into an embryo of a new mouse and let that new mouse develop. The idea is, since we've put in a TCR alpha and TCR beta, they will allelically exclude. And so all T cells that develop in this mouse will become clones of that original T cell that we've chosen. Typically, we will do this in a mouse that is also missing RAG2 because TCR alpha is so bad at allelic exclusion. I'm going to show you some data at the end here right now and at the beginning of the next lecture from a paper that did something like this that really helped us understand a lot of T cell development. That was before they knew about that TCR alpha allelic exclusion problem, and some of the data is quite messy in those figures. So you will see in a few places the data being a little messy. But if we were to make our TCR transgenic using this T cell that binds to influenza, and we were to insert those alpha and beta TCR genes into a mouse, a new mouse with the H2K background, and let those cells develop, if we looked in the thymus, we would see double negatives and double positives as the cells are developing. And then as you can see, we would only get CD8 single positives. We would see no CD4 single positives because we're only getting single positives that are sort of versions of that original T cell that we had. And if we look in the periphery, we'll see only CD8 T cells in this mouse. This mouse would, again, probably be quite unhealthy. It would not be able to live in the real world, only in a nice, clean lab environment. But we could use it for a lot of sources. We could use it as a source of cells, and we can use it to help us understand T cell development. So here you can see that if we inserted a transgenic receptor, an alpha and a beta chain that recognize MHC class 1, in the end, we would only get CD8 positive T cells. 100% of the peripheral T cells in that mouse would recognize whatever the antigen was that we put in. And with the MHC class 2, if we put in a transgenic receptor recognizing class 2, in the end, we would only get CD4 positive T cells to mature. Also remember that T cells are restricted by the MHC type of the thymus that they are developing in. So we could also think a little bit about what would happen if we were to make a transgenic receptor that bound to a particular type of MHC and then were to allow those T cells to develop because of the type of embryo that we inserted those genes into in a thymus of different MHC type. And so if we took a transgenic receptor that was restricted to MHC type A, and we put that alpha and beta T cell receptor gene into an embryo that is also of MHC type A, the cells would eventually develop in that MHC class A type thymus. They would get a signal, they would be able to be positively selected, and in the end, we would only get the single positives, the CD8s in this case, because it was an MHC class 1. If we were to put, however, that same transgenic receptor restricted to MHC class A, MHC type A, into an MHC type B embryo, the cells would try to develop. They would make that alpha and beta chain. They would allelically exclude and not be able to make any other alpha and beta chain choices, but they would be in a B type thymus. And so when those cells tried to get a signal for positive selection, all of those cells would be sad. They would never get their positive selection signal, and all of those cells would die by neglect. And in the end, if we looked in the periphery of those mice, we would see no single positive T cells, no T cells at all. Experiments like this are one of the ways we started to understand the role of MHC in T cell development and the order of some of the different types of populations that are seen in the thymus. And so you can see sort of another view of the same type of experiment here. If we were to take a mouse of the H2K MHC type, take our favorite T cell, this T cell that recognizes an influenza infected target cell, and it's a CD8 T cell, and clone out its alpha and beta TCR genes, and then put them into an H2K embryo, as shown on the left, we would see double positives, and those double positives would successfully keep developing and become single positive CD8 T cells. if we did the same experiment, but we put that alpha and beta T cell receptor gene into an H2D embryo and allowed that mouse to grow up, and we looked in that mouse's thymus, we would see that there were a bunch of double positive cells trying really hard to develop. They've all made the T cell receptor of the T cell above. They're trying to develop, but they're all going to die by neglect. And so in the end, no CD8 single positives will successfully be made in that mouse's thymus. I ran out of time here in class, and so I talked about some additional experiments that are classic experiments that helped us understand T cell development in the next class. And then I started on thinking about the movement of T cells around the periphery.